Hello everyone, welcome back to another session of regeneration biology. And in today's class, what we will study is organ regeneration, which is having its basics explored and also with some examples. What we should learn in this class is how and why some organisms are able to regenerate properly and some tissues are able to regenerate better than the other in the same species, and is there some hindrances that is occurring to the regeneration in some species, or by bypassing those hindrances, some stumble blocks, if you get rid of that, is it favoring a proper regenerative response? So that is what we are trying to learn. So organ repair and regeneration, it is an overview. So some questions can be asked easily. Will we ever be able to regenerate human organs and parts the way that a newt or an axolotl is able to do? Some very naive and interesting question. Our stem cells are able to reconstruct a whole damaged tissue or organ? While the answer at this point is premature, because we haven't reached a stage from cell we can make an organ from any species. The news, what we know, the future seems quite promising, good, when all possible strategies will be put in use. So that is what we can anticipate. What is, for example, the relationship of stem cells as we know them in mammals to the dedifferentiating newt cells? We know mammals have stem cells, we culture them, but dedifferentiated cells from newt or zebrafish or axolotl, are they similar in terms of their characteristics that create the source of regenerating tissue? Because in newt or axolotl or zebrafish, the existing tissue has to generate the so-called stem cells. Are there similarities? If so, can we learn from them how and why they were able to create or restore a damaged part in their body? Indeed, newt cells do express factors that characterize or give them the status of stem cells. Can we learn how the cells create these progenitor cells and guide them via scaffolds? Scaffolds are a technical term used to describe the extracellular matrix, or in other words, you are making a building, multi-storied building. The first what you make is pillars and beams, and then you fill the gap with the bricks. That is how you make rooms and houses. Same logic applies. So the pillars and beams are nothing but scaffold for a building. And we have to understand the scaffolds are essential, and these tissues only secrete the scaffold also. Okay, build tissues and organ scaffolds are essential. And can we artificially make some scaffold? Can we use one of the best scaffold possible so that the stem cells will be forced to make a damaged organ? So these are all some questions that remains. And there are three concepts. What we know is the classical animal regeneration and stem cell mediated and the tissue engineering. So classical animal regeneration means a damaged organ is regenerating by itself, not without too much of trouble or something. And then stem cell based. Can we use the stem cell and inoculate into a damaged area? Can they give rise to they can restore the damaged portion? That is another question. And third is a tissue engineering, where you are telling, okay, I don't need any support from the organism. I have stem cell, I have scaffold. I will make this organ on a petri dish and I will just transplant into the host. So these are all some dimensions which you should think about. So these three concepts can provide the necessary raw materials to materialize the final goals of regenerative biology and medicine. So when regeneration biology is used for a therapeutic purpose, we call it as a regenerative medicine. That is the word we use it. So if you look into the mammalian fetal organ regeneration, every animal, because we know mammals don't regenerate every organ, but whether it is a mouse or human or rat or any mammal you can think of, in the fetal stage, they have tremendous capacity of regeneration, and they do this without the formation of any scar. That is called scarless regeneration. Something which you can see in the model organism, axolotl or the newt larvae, etc. They do not give any scar. Scar means we have seen it already. It is the over deposition of fibroblast and the ECM. That is called fibrosis. Fibrosis is that what leads to scar. So if you have a surgical wound in your body, it is be seen even after 10-20 years you are able to see. That means that there was a scar. Scar stays lifelong, and it is nothing but the fibroblast and the ECM deposition which creates the scar. So in embryonic stage, that is the fetal, mammalian fetuses can regenerate skin and other tissues during early development, but this ability is lost as they mature. Once they mature, they lose this ability. You have several examples. Classic example is skin regeneration. Early fetal stage, fetal skin can heal rapidly and without scarring after injury. That is important. This process restores skin's strength, function, and extracellular matrix architecture is restored back. That is why there is no scar formation. If there is a damage, you can't find out where is the scar. Even if you notice, small kids, say 1-year-old kid, they are not in fetal stage. They are after birth only they will celebrate their first year. If they get some wound or injury on their skin surface, after some 1 or 2 years it will disappear. It's a slow process, but it will not stay. But if the same kid is getting an injury when they are 10 or 15 years old, that is going to stay rest of their life. So what you understand, 1-year-old kid versus a 10-year-old kid, something has changed. So late fetal stage, fetal skin heals with the scar formation. That is late fetal stage, similar to adult skin. So what you understand, the scar formation, and also you should understand one more thing, even if scar is formed in this late fetus, upon future several days or several months, the intensity of scar can come down. May not be in all, but it can come down, and it may reach a stage where it is not very conspicuous. So mammals fail to regenerate organs when wound contraction drives scar formation. Now you are learning a new terminology that is called wound contraction. So what is wound contraction? You have created, say, 1 square centimeter area, you made an open wound. So the cells or the organism is trying to close that wound as early as possible. And for this, migration of cells comes into picture, and then that locality, the ECM and also the cytoskeletal protein, the myofibroblast comes into picture, they close in. Because of this closing, the wound contraction happens. Like you may have seen it in rat has got 2 teeth, top 2 teeth, bottom 2 teeth, which roughly grows around 3 to 4 inches in a year, and it can penetrate through its brain. To avoid that, they will keep grinding stuff, and it will keep eroding the teeth so that it will stay in a peculiar length. Still, if you see a rat's teeth, front, top, bottom two, top 2, bottom 2 will be the longest teeth in spite of crawling. That's why they keep grinding stuff, not to eat, but just to sharpen their teeth. Why this has to be done? Because the bottom of this teeth is not contracting, unlike the rest of their teeth. Your teeth also, if you look your teeth very clearly, the base, the part which is joining the gum is contracted. That is why your teeth is not growing longer. Elephant tusk, it's nothing but teeth. It's continuously growing because there is no contraction. Why I gave this example is, contraction doesn't mean that there is no blood supply, no neurons, etc. It is there, but this contraction is good enough to prevent persistent growth. Means the nutrition supply is limited. So what you understand, when you have this contraction, the area is now getting into a sealing mode rather than going into a regeneration mode, because regeneration is always slow. It needs some open wound. If the wound remains open, and of course you don't want any infection to happen, it remains open, the regeneration can pursue. But the moment the wound is contracted, then it completely stops the propensity of formation of a neat wound closure or a neat repairing mechanism that leads to scar formation. So can we think of preventing the wound closure? That is how researchers have thought about it. Mammals in their early fetal stage have been shown to regenerate skin following severe injury. Early stage, no matter which mammal you are talking about. Investigators have been impressed by fetal wound healing observed in several mammalian species and have occasionally referred to it as flawless. Flawless means there is no scar formation, it looks perfect. While others have described the process as resembling regeneration. This scarless regeneration, even though it is skin, scarless regeneration or scarless wound healing is often mimics that of regeneration. So we can technically call it as a resembling regeneration scenario. Skin is a less complex tissue. It has got stem cell and also for the routine repair. But if there is too much of fibroblast deposit, then it will lead to scar formation. However, at about 2/3 of gestation, 2/3 of a gestation of a given species, depending upon which species you are talking about, 2/3 time has been passed. That is called a late fetal stage. The mammalian fetus experience a transition from the scarless skin wound healing to healing that leads to scar formation. Scar formation because of fibrosis. In sharp contrast to the late mammalian fetus or adult mammals, several amphibians, adult mammals and late fetuses have wound healing by scar formation. But several amphibians, say newt, say axolotl, can regenerate entire limbs spontaneously throughout their lifetime. Forget about scar, forget about wound healing, it generates the entire limb. We will learn about this much later in the coming classes, but keep this idea in your mind. Investigators have tried for several years to understand how early mammalian fetus heals without a scar to apply knowledge towards functional restoration in a diseased organs in human. Say there may be many people who are suffering because they don't have a hand or limb or a finger, etc., or a palm. If they can be restored, that is the $1,000,000 question. However, the reason behind the loss of regenerative ability during gestation remains unknown. After crossing the 2/3 of gestation, what stops or what blocks, what prevents the ability of that embryo to regenerate properly? And its regeneration is an intrinsic property or intrinsic pathway of mammals that cannot be changed later in life. If it has, it will stay. If it is not there, it is very difficult to rekindle. So this is a property which some organisms have, whereas the others don't. And even mammals have it in the early phases of their embryonic development in the fetal stage. Later it is been lost. So this poses a conundrum. That is why researchers are very curious what is so much different in post-delivery or late fetal stage. What is that is gone wrong? Scar formation is a key outcome of wound contraction. Like I told you, wound contraction is a adaptive response of the organism. You don't want any infection. You want to finish the job as fast as you can. You may have seen in National Geographic channel when a lion or a leopard that hunts, immediately it will try to export the food, the prey into a tree, or it will try to eat as fast as you can. Because scavengers like a hyena can come or a larger predators can come and snatch away its food. Same logic applies. The wound healing happens as fast as possible, infection can be avoided. If you have an open wound, it is a welcome signal for a lot of pathogens to come and infest, and you can get into septicemia and the organism may die. You may wonder, it may not be a serious thing in modern world where you go to hospital and get it fixed. But in olden time, as a hunter and gatherer, or when you are an uncivilized as a species, a wound can give you a death sentence. So organism with a fastest wound closure, or wound contraction, even though there is a scar formation, that is not an issue. In contracting adult skin wound, many MFB, what is MFB? Myofibroblast, are held together in assemblies by intercellular junctions. Many myofibroblasts comes into picture and individual cells are connected through, just like 10 people are standing when they are crossing a river, they are holding their hands so that they act like a massive organism, hence they will not fly away or flew away into the water flow, something like that. The organization of such myofibroblast assemblies, particularly the spatial orientation of individual MFB axis, appears to align with the physiological macroscopic wound contraction forces that eventually reduce the wound size. Take home message is, when individual cells keep coming and they establish a connection And because of this connection, they being myofibroblasts, they have the contractile property, so they will come much faster. If I want to give an example, say you put 10 nails in a circle and you put a rubber band. And if the rubber band is tighter and tighter, it will try to bend the nails, and rubber band is trying to come into the center. So the nails, vertical nails now will slant, and the rubber band is dragging it. Something of that logic applies. Myofibroblasts will try to, although cells are needed, but they will try to stretch it. So this stretching will create lots of fibrosis and scar formation. The spatial distribution of MFB assemblies and their resulting mechanisms, it will create a stress field generated in the injury site due to the contractile forces applied by the MFB or myofibroblasts depend on the anatomical shape of the injured organ and the local geometry of the wound undergoing healing. If it is a circle, it will come contractile manner. If it is an oval shape cut or a square shape cut, accordingly the wound closure also will happen. Not that it always has to be circular. This picture explains everything quite well. Wound contraction and associated wound healing responses in 3 adult injury models in the absence of spontaneous healing and the presence of a dermal regeneration template, that's called a DRT graft. DRT graft in a simplistic sense, I can tell you that contraction is prevented. That means wound is trying to contract and you are putting a DRT graft in there, it is preventing. It is not letting this wound contraction to happen. Rather, in other words, you are delaying the healing process. In other words, if you can explain. So you can see here, blocking of wound contraction by DRT is been done in different tissues. It has been done in skin, it has been done in peripheral nerve, it is also done in conjunctiva. That is the boundary between your cornea and the sclera. Cornea and the the adjacent white part of your eye, that is conjunctiva, and there also it has been tried multiple places. So what is the simplistic form is a simple and direct test of the hypothesis that wound contraction, you are asking the question is wound contraction, if you prevent it somehow, are we getting a better regeneration? That is the question. So we are thinking of a scenario, can we put a dermal regeneration template, which is a structure which do not allow the wound contraction. That means it will retain the open wound as open wound. Like if you have a wound, doctors will put a ligature, they will put a suture. That means they are bringing the wound, two wounds closer. Now you are retaining the wound as open as possible. Okay, that is the scenario. And associated, and also this dermal regeneration template also have got a scaffold, which means it is providing the ECM, extracellular matrix. Wound contraction is associated with the scar formation, consists of using an efficient contraction blocker, that is the DRT, and observing if such use of such a DRT prevents the scar formation. That is the question. Dermis regeneration template, a highly porous collagen based scaffold, that is called a DRT. Its original material is ECM protein, that is collagen, and effectively blocks contraction in skin wounds and peripheral nerve wounds. And also it is done in conjunctival after stroma, damage to the conjunctiva. If any damage happened to your cornea, you can go blind, but if damage to the conjunctiva, it can heal. DRT induced downregulation of the inflammatory response. So another important thing you should understand is the DRT, when present, it reduces the inflammatory response, because absence of a proper collagen can invite inflammatory response. But if you put a DRT, the inflammatory response is compromised. This is something you should keep in mind, because this subject is going to come after a few slides in this class. And it can reduce the downregulation of the inflammatory response. Inflammatory response is done by the immune system, and the inflammation means, like if there is a mosquito bite, you have a pimple, you know that surrounding will be reddish in color. That is simple term inflammation means, and this DRT reduces it, including cytokines required for MFB differentiation, such as TGF-beta1, could account for this healing benefit. So the presence of low inflammation, presence of a proper required scaffold, and the absence of wound contraction favor a slow healing but scar-free healing. So this is what has been shown in these different tissues. Like skin, you can see tissue outcome. One is no DRT, natural scenario, there is a scar, whereas with the DRT, the scar is minimal because less ECM deposition. And no DRT, you can see this is what you see in peripheral nerve, whereas with the DRT, you see proper nerve formation here. And here no DRT, in conjunctiva, you end up getting a too much of deposition of the matrix or collagen, whereas with the DRT, you see a much milder tissue or milder wound healing in the conjunctiva as well. So emerging rules for inducing organ regeneration is something very interesting. In regenerative medicine, no widely accepted paradigm is currently available to guide the formulation of new theories on regeneration mechanisms in adult. No prevalent or accepted universal theories exist. Three structural features of scaffolds that are required for the regenerative efficiency. Like we saw, DRT is acting as a scaffold and preventing the wound closure. So an active scaffold emerges as a temporarily insoluble collagen surface. That is what usually happens, equipped with sufficient ligands. So this scaffold should also provide ligands. Ligands means that can favor the migration of cells. You don't want some meshwork is being deposited. You want them to be friendly to the cells which is migrating, and they should have sufficient ligands for integrins of the contractile cells, the cells which are able to migrate over it, that inhibits wound contraction. So that is what is needed. Scaffold, DRT scaffold should be there, and they should also have ligands that do not allow this contraction to happen, while also serving as a topographic template for new stroma synthesis, because cells are blind. They don't know where to go, how to go, and which place to settle it down. So they depend on this scaffold or this so-called ECM. So once you artificially put it, they are not in a tension of randomly coming or randomly closing the wound, and there is no contraction of the wound is necessary because of the presence of this artificially put in scaffold, and this helps in the healing without scar formation. And decellularized matrices. So many times what the people do is they take a tissue, remove the cells, kill the cells, and protein stays back. And that is called a decellularized matrix, and it will look like a cotton candy. All of you would have seen what is cotton candy, which is made of sugar, sucrose, but it has got holes. That is why it is looking like puffy. So if you decellularize, the liver will look like a big cotton candy, but it will be light in weight. No cells are there, but only the ECM proteins are there. Matrices have been used during the past few years to regenerate the whole or in part the urethra, abdominal wall, Achilles tendon, the bladder, the trachea, and other organs in several animal models and occasionally in humans. People have artificially created a urinary bladder by this decellularized methods and transplanted into humans also. So they are all been successfully done because urinary bladder is one of the easier structure. So organ repair and regeneration, an overview. Several organs have intrinsic ability to regenerate, a distinctive feature that varies among organisms. Organ regeneration is a process that is not fully understood as of now. However, when its underlying mechanisms are unraveled, it holds tremendous therapeutic potential for humans. That is why people look into all possible ways. Can we tweak the regeneration? Can we tweak the wound healing? Can we think of making an organ, etc.? Repair and regenerative potential of various organs and organ systems have been explored, such as the thymus, the adrenal gland, thyroid gland, intestine, lungs, heart, liver, blood vessel, germ cells, and nervous system, eye tissues, hair cells, kidney, and bladder. Bladder means urinary bladder, skin, hair follicles, pancreas, bone, and cartilage. None of the tissues have been spared. Pretty much every organ has been actively explored and with some good success in many of these system. So organ repair and regeneration, if you look further, you can see in this picture, there are three sections, A, B, and C. So in A, you are seeing local cells either differentiated hepatocytes or tissue-specific stem cell. This is the liver is an example, and proliferate and repopulate the injured area, providing essential growth factors. So the so-called scaffold should provide this required growth factors. This concept can be applied to the dedifferentiation of cells in the injured site, what we see in the amphibians usually. And in B section, what you are seeing, an artificial scaffold. This is natural. This is an artificial scaffold is seeded with the cells to repopulate and deconstruct the lost part of the organ. So you have a scaffold. In scaffold, different spots, random distant spots, equidistant spots, you are inoculating one cell, and these cells will divide and they will colonize. That is the whole concept. The cells from the bone marrow, this part C, you can see, can be brought via blood vessels to the injured area and contribute to the repair or regeneration. So this is what the whole concept is. You can learn from liver, you can use artificial scaffold, and you can also learn from the bone marrow stem cells. So now let us see how the inflammation and immunity is coming into picture for an organ regeneration. Like in the some time ago, we discussed that the inflammation and immunity can be a problematic thing. So let us see, is there any evidence to support this? Both inflammation and its resolution are necessary for the successful regeneration of amphibian limbs and zebrafish caudal fins. Inflammation is must. If inflammation is not there, you are preventing the inflammation, forget about regeneration. You can't get it. So that means inflammation is a welcome signal. Regeneration requires immigrating macrophages to remodel the ECM, extracellular matrix, and possibly resolve the inflammation. Inflammation happens, then macrophages comes into picture and they try to deal with it. Macrophages are of different types, M1 and M2 are there. We will see them in the future class about those things. And numerous pro-resolution genes are expressed in the early phase of regeneration. In the tissue, the damaged tissue, along with this inflammation, they also will trigger the expression of some of the genes that is allowing that resident cells to dedifferentiate. That is what we call it as resolution specific genes. Resolution of inflammation is required for pattern related gene expression. So you have to get rid of this inflammation. Inflammation is needed to start with, and then you don't want that inflammation to stay back. You want the inflammation to go off, and then only you can have the regeneration specific gene expression. So immediate amputation, you see an immediate early gene response which is associated with inflammation. Then inflammation should go off. Then you start getting some proper dedifferentiation specific gene expression. And prolonged inflammation prevents patterning and regeneration. So what you understand, you do not want a prolonged regeneration. Say you went to a restaurant for having meal at breakfast time, but you are not coming out of the restaurant. You are constantly eating. Is that good? So you go to a restaurant, have one plate of food, finish it, come out and do your job, what is needed. So that is why inflammation is must, but you don't want inflammation to continue. It can affect the patterning and the regeneration. Evidence supports the role of regulatory T cells and the suppression of autoimmune reactions in stimulating regeneration in incompetent aneurin larvae. So what people have done it, if you can make an aneurin larvae regeneration incompetent, normally frogs, after their metamorphosis, their regeneration is compromised. They are not able to regenerate. Like if you see a frog, the larvae can regenerate better, but the adult frog cannot regenerate effectively. It can do wound healing, but it doesn't regenerate effectively. But if you compromise its immune system by blocking some cell immune response, etc., then you can get a better response. So in this picture, what you can see is expression profiles after amputation is the immune related expression of chemokines, cytokines, and annexins, etc. The major type of leukocytes involved are being depicted here. As you can see in this graph, pro-inflammatory cytokine and then anti-inflammatory cytokine. During this time, neutrophil comes into picture, macrophages comes into picture. Then during this time, cellular reprogramming happens and growth and organ patterning is happening. So we should understand in immunosuppressive method in Xenopus larvae favoring regeneration, and this dedifferentiation is necessary for which is facilitated by the inflammatory response. We will learn more about regeneration in the next class. Thank you.